Hello everyone. In this video we are going to talk about why is cultivation required, methods of inoculation, advantages of solid media, advantages of liquid media, common staining techniques, gram stain, acid fast stain. Cultivation is required for the isolation of organisms in pure culture from the clinical specimens and their final identification. For antimicrobial drug sensitivity tests, preparations of the antigens to be employed in serodiagnosis of infective disease. For the preparation of the vaccine, methods of inoculation, streak culture. It is the routine procedure for bacterial isolation in the pure culture. The surface of a semi-solid medium in a petri dish may be inoculated with a specimen by several methods. The primary inoculation may be made by a loop, swab, or other suitable device. Low or carpet culture, also called as antibiotic sensitivity testing. Bacteriophage typing, uniform surface growth, used for the preparation of bacterial antigens and vaccination. Stab culture, it is performed by 4-inch long thick straight wire charged with culture material deep inside the agar. Pour plate culture method, several tube containing 15 ml agar, molten agar is cooled to 45 degrees Celsius, is poured into petri dish containing a specific amount of diluted samples. Advantages of inoculation on solid media. There is separate colony formation. Identification of most bacterial specimens can be made. Isolation of bacterial count and relative proportion of different bacterial species can be made. Isolation of bacteria in pure culture can be made by picking isolated bacterial colonies and subculturing into fresh medium. This is necessary for full identification of bacteria. Inoculation on liquid media. Advantages. When bacteria are present in small numbers in the inocula, they will grow only in liquid media. For example, blood culture. Liquid media are widely used for biochemical tests. Large inocula can be tested in liquid media. For example, gauge for sterility test. Increased sensitivity compared with solid media, especially for CSF, pleural fluid, biopsy, etc. Anaerobic culture methods. Anaerobic bacteria require incubation without oxygen. Methods of anaerobiosis. By displacement of oxygen. Cultivation in vacuum. Displacement of oxygen by inert gas like hydrogen or nitrogen. Absorption of oxygen by chemicals. By biological methods. Common staining techniques. First one is simple stain. Watery solution of single basic dye such as methylene blue are used as simple stain. Second one is negative staining, also called as capsule stain. The negative stain is particularly useful for determining cell size and arrangement. It can also be used to stain cells that are too delicate to be heat fixed. Negative staining stain background, not bacteria. Impregnation method. Bacterial cells are too thin to be seen under microscope are visible by silver impregnation on their surface, used for the demonstration of bacterial flagella and spirochaetes. Differential stains. They impart different colonies to different bacteria or their structures. Gram stain. It is most widely used stain in medical bacteriology. The stain was discovered by Christian Gram. Methods. Smear of specimen or bacterial culture is made with crystal violet or methyl violet solution for 1 minute. Pour off crystal violet and add dilute solution of iodine such as grams iodine and keep it for 1 minute. Wash with water. Decolorization with alcohol for 10 to 30 seconds. Wash again with water. Counter stain with dye containing color for 20 to 30 seconds. Observation. Gram positive bacteria stain violet. Gram negative bacteria stain pink. Gram positive bacteria are all cocci except Neisseria and Branhamella. Gram negative bacteria. All bacilli are gram negative except corynebacterium, mycobacterium, bacillus, Clostridium, Actinomyces, Listeria. Acid fast stain was discovered by Ehrlich and was modified by Zill and Nielsen. It is extremely difficult to stain mycobacterium with ordinary stain like gram stain because it contains lipid which makes decolorization. So it can be done by acid fast stain but requires fluorescein. Procedure. Smear should be made, then added strong carbon fuchsin for 5 to 10 minutes, then wash with tap water. Decolorize with acid alcohol mixture till get a pink color in a smear for 3 to 5 minutes, then wash. Add methylene blue for 2 minutes, then wash. Allow to dry and focus under microscope. Observation: acid-fast bacilli appears red in blue background of pus cells and epithelial cells.